Welcome to Roadmap to Residency, Evidence-Based Application. In this video, we will be talking about bacterial growth and replication. So when you see the bacterial growth curve, okay, now there are different sort of phases in case a bacterial growth curve. Generally, three important phases. First one is the lag phase. Then there comes the log phase here. Then there comes the stationary phase, and then there comes the declining phase. So first, in case of lag phase, what happens is that now the bacteria is getting acclimated to the environment. The bacteria has just entered the cell and it is getting acclimated to the environment and all. In case of log phase, now the bacteria is rapidly dividing in case of log phase. The number of bacteria is rapidly increasing in case of log phase. It is rapidly consuming the cell components, the energy and all. Now, in case of stationary phase, here the cell's energy have been used up and the bacteria have stopped multiplying. And ultimately, after some time, what happens is that since there is no energy in the cell, since there is nothing to eat for bacteria in the cell, then what happens is that the bacteria starts dying, and that is called as the declining phase. So we have the four phases: lag phase, log phase, stationary phase, and then there comes the declining phase. Now, whenever the bacteria multiplies, then obviously there should be gene transfer. So now we will be studying about the different modes of gene transfer. So bacteria often transfer genetic material, which is the key for evolution of antibiotic resistance. There are three main mechanisms behind the gene transfer of bacteria: transformation, conjugation, and transduction. Bacterial transformation is when there is direct uptake of DNA from the surrounding environment. It allows for the evolution of DNA over time, and it is very useful technique in microlabs. It introduces genes to bacteria for replication. Bacterial conjugation is when there is transfer from one cell to another cell of bacteria via the pilus. It is through the physical contact between the two organisms, and DNA is transferred via plasmids. Now, plasmids is a small DNA molecule within a cell, which is physically separated from the chromosomal DNA, and they can replicate independently. They can contain genes for antibiotic resistance and toxins, and they can be transferred from one bacteria to another. Now, let's review the first part of the bacterial genetics. So, transformation is when the competent bacteria combine and import short pieces of environmental naked bacterial chromosomal DNA. The transfer and expression of newly transferred genes is called as transformation, and it is a feature of many bacteria, which is maybe Strep pneumonia, Haemophilus influenzae, and Neisseria. Now, talking about conjugation. Now, conjugation is when the F plus plasmid contains genes required for sex pilus and conjugation, and bacteria without this plasmid are termed as F minus. Okay, just remember X and Y. So now the sex pilus of the X, that is the F plus bacterium, contacts the F minus bacterium through formation of a sex pilus, which acts as a mating bridge. And now the single strand of plasmid DNA is transferred from the F plus cell to the F minus cell. Okay, now since the both of the cell contains the DNA, now the both have become F plus cell, and after that there is no further transfer of chromosomal DNA. Now, F plus plasmid can become incorporated into the bacterial chromosomal DNA termed high-frequency recombination cell or HFR cell. Transfer of leading part of plasmid and a few flanking chromosomal genes occur, and it is a high-frequency recombination which may integrate some of those bacterial genes. The recipient cell remains F minus, but now may have new bacterial genes because the leading portion of plasmid transfers along with flanking bacterial component. So the plasmid and bacterial component both have transferred. Now talking about the transduction, which can be either generalized transduction or a specialized transduction. So generalized transduction is when the packaging error takes place. That is when the lytic phase infects the bacterium, leading to the cleavage of bacterial DNA. And then the part of the bacterial chromosomal DNA may become packaged in the phase capsid. Here, the phase infects another bacterium and transferring these genes to another bacterium. So just remember, like a mosquito is there. Mosquito sucks the blood from one human being, and then again it goes to bite another human being, and then there is the transfer of viruses and bacteria. So just remember bacteria phase as some mosquito of different bacterial cells. Specialized is an excision event. When the lysogenic phase infects the bacterium, the viral DNA incorporates into bacterial chromosome, and when phase DNA is excised, the flanking bacterial genes may be excised with it. So you can see the viral DNA here, and this is the bacteria. So the viral DNA incorporates in the bacterial DNA. And again, when the phase particles carry the bacterial DNA, that is, it can divide it into this inside the cell. And when there is release of new phase from the live cell, it infects another bacteria. Now this new phase will infect another bacteria, and again the genes are different from the donor and recipient. So it is completely different gene, and it is shown by some organisms like uh, botulinum toxins, cholera toxin, diphtheria toxin, and Zika toxin. So generally, the genes for the following five bacterial toxins are encoded in the lysogenic phase, and it is very, very important to remember these names, the toxins which are faced by the lysogenic phase. Straightforward question will come to asking you about which of the toxin gets formed by the lysogenic phase reaction. Now, transposition is a new concept. So transposition is a jumping process involving a transposon, which is a specialized segment of DNA, which can copy and excise itself and then insert into the same DNA molecule or an unrelated DNA. It is critical in creating plasmids with multiple drug resistance and transfer across spatial lines. Now, talking about the binary fission. Now, binary fission occurs in the primitive organism. So, yeah, it is a very primitive way of division of bacterial molecules. Now, the cell replicates this DNA here, and the cytoplasmic membrane elongates, separating the DNA molecules, and then the cross wall forms between the membranes, and then through the complete formation of cell wall, it forms daughter cells, and here you have two new bacteria, and again the process goes on. So this is a very primitive method of of the organism to divide. Here we can see one question with us. So in which of the following locations did this mutation most likely occur? So obviously, when there is growth, when there is transfer of DNA, when there is transfer of genetic material, then obviously there may be some sites, or there may be some event when there may be some mutations occurring on. So this question will just talk with you about how the mutation occurs in the DNA or in the bacteria and all. So microbiologists are investigating sugar metabolism in wild type and mutant strains of E. coli. Both strains are found to grow viable colonies on lactose-containing media. Each strain is then cultured on a new growth medium containing only glucose. Representative colonies of each strain from the new media undergo Western blot processing using a fluorescently labeled probe specific for beta-galactosides. Wild-type bacterial colonies are found to contain only trace quantities of beta-galactosides. However, the mutant colonies express significant amount of beta-galactosides. Further analysis reveals that the variant strain contains a mutation that inhibits the binding of a certain protein to its regulatory sequence. Now, in which of the following locations did this mutation most likely occur? Activated protein binding site, operator locus, promoter region, promote activated protein CAP gene, and RNA polymerase cistron. So the lac operon is regulated by two distinct mechanisms, negatively by binding of the repressor protein to the operator locus and positively by CAMP-CAP binding upstream from the promoter region. Constitutive expression of the structural genes of the lac operon occurs with mutations that impair the binding of the repressor protein to its regulatory sequence in the operator region. Thank you for watching. Keep studying hard.